हॅलो फ्रेंड्स, राणा अकॅडमी सर्वांचं स्वागत आहे. आपण स्टार्ट केली आहे 11th क्लासची बायोलॉजी. हा दुसरा चॅप्टर आहे. पहिला चॅप्टर आपण पाहिला आहे ह्याच्या अगोदर. चला तर मग व्हिडिओला सुरुवात करूया. तुम्ही या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला सबस्क्राईब करून ठेवा, जेणेकरून तुम्हाला पुढचे व्हिडिओ पण पाहायला भेटतील आणि पाठीमागचे पण अपलोड केलेले तुम्हाला भेटतील एका सिरीज वाईज. तर चला तर मग सुरुवात करूया व्हिडिओला. बायोलॉजी 11वीची महाराष्ट्र स्टेट बोर्डची दुसरा चॅप्टर, सिस्टमॅटिक्स ऑफ लिव्हिंग ऑर्गॅनिझम्स. There is a great diversity of organisms around us since time immemorial. Where humans have been exploiting this wealth for our own benefit. During this process, man tried to differentiate between the and identify the organisms. This evolved into the branch of biology known as classification that has led eventually to systematics. The methods of classification dates back to ancient time. Have Indian, Greek, and Roman philosophers have contributed their might be to systematize science. Systematics. Systematics is the study of kinds of diversity of organisms and their comparative and evolutionary relationship. G. Simpson, 1961. Taxonomy. Taxonomy means classification following certain rules or principles. Word taxonomy comes from two Greek words, taxis meaning arrangement of, nomos meaning law or rule. The term taxonomy was first introduced by A.P. de Candolle, Swiss botanist, 1778 to 1841. Classification. It is arrangement of organisms or group of organisms in distinct categories in accordance with a particular or well-established plan. This classification is based on similarities and dissimilarities among the organisms. Artificial. It is the classification that is based on few easily observable and non-evolutionary features that as habit, color, etc., often irrespective of their affinity relationship with other organisms. Example, linear system of classification. Natural. It is the classification which is based on objectivity, significant rather than being selected for convenience, like artificial system of classification. Example, Bentham and Hooker's system of classification. Phylogenetic. It is the classification based on common evolutionary descent. Example, Engler and Plantal classification. In the system of classification, the terms like taxa and categories are often used. Each category is referred as a unit of classification. In fact, it represents the rank and is commonly termed as taxon. Bacteria, archaea, eukarya, common ancestor. Phylogenetic descent. Three domains of life. It is believed that the life originated on Earth in its very simple form. Constant struggle of the early living beings gave rise to more and more perfect forms of life. This struggle and progress in evolution, which led to formation of diverse life forms. Carl Lewis in 1990 proposed three domains of life to classify life forms. They are archaea, bacteria, and eukarya. Domain is as a unit larger than kingdom in the system of classification. Bacteria and archaea both have prokaryotic cells, whereas eukarya have eukaryotic cells. The three domains have very unique ribosomal RNA. Archaea are known for their survival in very extreme conditions like high temperature, salinity, acidic conditions, etc. Bacteria, through prokaryotes, differ from archaea in structure of cell wall. Chemotaxonomy. It is a method of biological classification based on similarities and differences in structure of certain compounds present among the organisms being classified. In short, it is the classification based on chemical constituents of organism. Example: archaea, cell wall is without peptidoglycan, and that of prokarya is with peptidoglycan. Among eukarya, fungi have chitinous cell wall, while plants have cellulosic cell wall. Numerical taxonomy: the system is based on quantification of characters and development in algorithm for classification. The basic aim of this taxonomy was to create a taxonomy using numeric algorithmic like cluster analysis rather than using subjective evaluation of their properties. This system was first proposed by Sokal and Smith in 1963. Cladogram: it is a typical branching pattern as shown on previous page. A diagram of three domains of life is a cladogram. It represents a hypothetical relationship denoting a comparison of organisms and their common ancestors. Phylogeny: it is evolutionary relationship of all organisms. It is an important tool in classification of as it takes into account not merely the morphological status but also the relationship of one group of organisms with other groups of life. The system helps to understand the evolution and also focuses on the similarities of their metabolic functioning, whose three dominant concept as well as Whitaker's five kingdom classification, five kingdom system are very good example of phylogenetic relationship. DNA barcoding. DNA barcoding is a new method for the identification of any species based on its DNA sequence from a tiny tissue sample of the organism number under study. It helps to study newly identified species as well as understanding ecological and evolutionary relationships between living beings. The process of DNA barcoding includes two basic steps: collecting DNA barcodes, data of known species, and matching the barcode sequence of the unknown sample against the barcode library. For identification, DNA barcoding has many applications. A few to mention are protection of endangered species, preservation of natural resources, pest control in agriculture, and identifying disease vectors, and authentication of natural health products, and identification of medicinal plants. Taxonomic categories. Classification is not a single step process but involves a hierarchy of steps in which each step presents a rank or category. Since the category is a part of overall taxonomic arrangement, it is called a taxonomic category, and all categories together constitute the taxonomic hierarchy. Kingdom, division, class, order, family, genus, species are the categories of hierarchical sequence. These are compulsory categories. Besides, there are some facultative categories like suborder, subfamily, etc. to be used as per need. Taxonomic hierarchy. Taxon. A taxon is a taxonomic group of any ranks in the system of classification. H.J. Lamb, 1948. Example: in plant kingdom, each of one of the meaning following is such as angiospermia, dicotyledonea, polypetalia, malvasia represents a taxonomic group, that is a taxon. Category: taxon. Kingdom: Plantae Animalia. Division: Angiosperms chordata. Class: Dicotyledon Reptilia. Subclass: Polypetalia diapsida. Series: Thalamiflorea. Force. Order: Malvasia squamata. Family: Malvasia elapidea. Genus: Hibiscus nasa. Species: NASA Sinensis nasa. Units of classification: species. Species is the principle of natural taxonomic unit ranking below the genus and denoted by Latin binomial, considered as the basic unit of classification. It is the group of organisms that can interbreed under natural conditions to produce fertile offspring. It was thought to be an individual, stable, and static unit. However, in the modern taxonomy, subdivision of species such as subspecies, varieties, and populations are seen and given more importance. Genus is the taxonomic rank or category larger than species used in the biological classification of living and fossil organisms. Genus is a group of species bearing close resemblance to one another in their morphological characters, but they do not interbreed. Example: tiger, leopard, lion. All three belong to same genus Panthera. They have belong to some genus Panthera. They have common characters. Eight are different from each other because their genus is same but species is different. Another example of genus Solanum, brinjal and potato both belong to this genus family. It is one of the major hierarchical taxonomic rank. A family represents a group of closely related genera. Example: genera like hibiscus, gossypium, Seda, Bombax and are included in same family. Malvasia cat also belong to family of leopards, tigers and lions. Family Felidae, but dog belongs to different family Canidia order. It is a taxonomic rank used in the classification of organisms and recognized by nomenclature codes. An order is a group of closely related families showing definite affinities. Now, order thus is the step above family in taxonomic hierarchy. The members belonging to same order but different families may show very few dissimilarities. Example: family Papaveraceae, Brassicaceae, Caparidaceae, etc. with parietal placentation are grouped in order parietals. Families of dogs and cats too are different. They belonging to same order Carnivora class. The class is the distinct taxonomic rank of biological classification having its own distinctive name. A group of higher taxonomic rank than an order. Class is the assemblage of closely aligned orders. Assemblage of closely allied orders. Orders Carnivora and primates belong to class Mammalia. Thus, monkeys, gorillas, gibbons, the primates, and dogs, cats, tigers, carnivora belong to same class. Division phylum. The division is the category composed of related classes. Example: division Angiospermia includes two classes, Dicotyledonea and Monocotyledonea. In animal classification of division is the unit of category phylum. Subkingdom. Different divisions having some similarities form subkingdom. Example: the divisions Angiospermia and Gymnospermia will the subkingdom Phanerogams and Supermetaphyta kingdom. It is the highest taxonomic category composed of different subkingdoms. Example: subkingdom Phanerogams and Cryptogams form the plant kingdom or plantae, which includes all the plants, while all animals are included in kingdom Animalia. The taxonomic category we have considered so far are broad categories. Scientists have added subcategories to this in order to place organisms in more scientific manner. You will observe that as we go higher and in taxonomical ladder, number of common characters go on decreasing. If we are comparing two organisms that are related to each other all only at division and phylum level, their classification may become difficult. Nomenclature. Any object that becomes known to human intelligence must possess a name. It may not be possible or convenient or to describe it in order to communicate ideas about it. The art of naming the objects is in fact the science called nomenclature. All living organisms are known by a particular name. Vernacular, local name, common name. Widely distributed organisms have a large number of common names. Pansy grown in most European and American gardens has about 50 common English names. In a multilingual country like India, almost all useful plants have local names which differ from language to language and even from dialect to dialect. As in Ayurveda, Mangifera indica is known by over 58 different names, all in the Sanskrit language. Hence, the common names obviously have limited uses of, and for universal applications, a unique name for particular individual is very much essential. Scientific names to overcome the difficult raised by common names. Scientists have given scientific names to all known organisms. These are systematic, thus provide means for international communication. Initially, the polynomial system was used, but Carl Linnaeus used a binomial system of nomenclature. He introduced this system in this book, Species Plantarum, published in 1753. International Code of Botanical Nomenclature (ICBN) has been set up of confirm all the scientific names. Before 2011, the code which was set up to confirm the scientific names was ICBN, means International Code of Botanical Nomenclature. Recently, a National Botanical Congress (ICB) was held in Shenzhen, China, in July 2017. This code is also called Shenzhen's code, and so the old code ICBN has been changed to ICNAP, which means International Code of Nomenclature for Algae, Fungi and Plants. This code was published on 26 June 2018. According to this system, the scientific name of sunflower is Helianthus annux. Then the above Helianthus indicates names of the above names of the genus, and second word anus denotes name of the species. The binomial nomenclature system follows certain rules. Name of organism is composed of two Latin Greek words. Genetic epithet is a simple noun. It should come first and begin with capital letter. A specific epithet is the descriptive adjective which should come later and begin with small letter. Scientific epithet must be underlined separately if handwritten and must be printed in italics. The generic and specific epithet should not have less than 3 letters and more than 13 letters. Usually the name of author who names a plant or animal is also written in full or abbreviated form after scientific names. Example: Mangifera indica L, where L stands for Linnaeus. Known the scientist Carl Linnaeus classified living organisms in two kingdoms based on the mode of nutrition, whether they are the migratory, sedentary, etc. But this broad classification as kingdom planting and kingdom Animalia area was found inadequate. It could not classify the organisms that show now characters of both the kingdom. For example, bacteria, fungi, euglena, etc. Hence, to avoid confusion, scientist R.H. Whitaker in 1969 proposed five kingdom system of classification. This system shows the phylogenetic relationship between the organisms. The five kingdom are kingdom Monera, kingdom Protista, kingdom Plantae, kingdom Fungi, kingdom Animalia. Excellent features of the five kingdom Monera. It contains unicellular organisms with prokaryotic unicellular organisms. Monera includes unicellular prokaryotic organisms. These are omnipresent. They are found in oil. They are found in all types of environment, which are not generally photoautotrophs, which are not generally inhibited by other living beings. Few are photoautotrophs and chemoautotrophs, but majority of heterotrophic in nature. These organisms do not have a well-defined nucleus. DNA exists as the simple double stranded circular single chromosome called as nucleoid. Smaller circular micromolecules of DNA as extra chromosomal genetic elements called plasmids are often present. Cell wall is made up of peptidoglycan, also called murin, which is a polymer of sugars and amino acids. Membrane bound organelles, example mitochondria, chloroplast, endoplasmic reticulum are absent. Ribosomes are smaller in size 70s than in eukaryotic cell. The mode of reproduction in Monera is asexual and or with the help of binary fission or budding. Very rarely sexual reproduction is by budding. Very rarely used sexual reproduction is by conjugation method. Morphologically, now bacteria are categorized into four groups: the spherical coccus, the rod-shaped bacillus, the comma or kidney-shaped vibrio, and the spiral-shaped spirillum. On the basis of evolution, bacteria can be classified as archaebacteria and eubacteria. Different shapes of bacterial cells. Figure 2.3. Archaebacteria, these are differentiated from other bacteria on the basis of their different cellular features. These bacteria are mostly found in the extreme environments, hence termed extremophiles. They are found in a variety of places, from volcanic craters to salty lakes and hot springs. Their ability to withstand such hostile environments speaks of their capacity that can withstand high salinities are called halophiles, while those that withstand extreme temperature are known as thermophiles. Very common example is of methanogenic bacteria found in gut of ruminants, cows, and buffaloes. These bacteria help in production of methane in biogas plants. Eubacteria. These are commonly referred as true bacteria. They have cell wall of peptidoglycan. They are found as autotrophs and heterotrophs. On the autotrophs can be photosynthetic like chlorobase, Greek sulfur bacteria, and chromatium or chemosynthetic like sulfur bacteria. These are mostly multicellular filamentous forms living in fresh water. The body is covered by mucilaginous sheath. The genetic material is typical prokaryotic, say chlorophyll A, chlorophyll B. Carotenes and xanthophylls are the photosynthetic pigments. Filaments show heterocysts, which help in nitrogen fixation. Heterotrophs are the most abundant. Most of them are decomposers and nodes. known for breaking down large molecules in simple so molecules or minerals. They can be anaerobes, helping in curdling of milk, lactobacillia, fixation of nitrogen, azotobacter, antibiotic production, streptomyces, the compositing, the degrading oil. But the story does not end here. Some of them are pathogens that is causing disease, thyroid, chlorella, tuberculosis, tetanus, cyanobacterium, snowstorm. Mycoplasma, these are smallest of the living forms. They do not have a cell wall. Many forms are pathogenic. They are found resistant to common antibiotics due to the absence of cell wall. Kingdom Protista. This group includes all the multicellular, unicellular, and eukaryotic organisms. These organisms show link with all eukaryotic kingdoms like plant, fungi, and animalia. Plant like Protistas. They are also termed chrysophytes. They are commonly termed phytoplanktons. They are microscopic and mostly photosynthetic and are major producers in oceans. Most of them are referred to as diatoms as they have body wall made up of two soap box like fitting silica covers. Diatomaceous earth has nothing but these shells left behind for so many years. Diatomaceous earth is granular, hence finds use in polishing and filtration diatoms. In animal like protistas, they are also termed protozoans. They lack cell wall. They are heterotrophs. They are believed to be primitive animal forms. Amoeboid protozoans have pseudopodia as locomotory organs. The amoeba is free living forms, but antamoeba is endoparasite and causes amoebic dysentery. Flagellated protozoans have flagella as locomotory organs. Trypanosoma is a common flagellated pathogen which causes sleeping sickness. Paramecium is a ciliate protozoans having cilia for locomotion of in paramecium gullet, a cavity open on the cell surface. Plasmodium is a protozoan protozoa. It causes malaria. It forms spores in one of its life stage. Dinoflagellates. These are aquatic, mostly marine and photosynthetic. The cell wall possesses a pair of flagella. They have a wide range of photosynthetic pigments which can be yellow, green, brown, blue, and red. Gonialex and dinoflagellate that is responsible for famous red tide. It makes even sea appear red. Fungi like protistas, and they are commonly called from the group Myxomycetes. These are of saprophytic organisms found on decaying living leaves. Their cells aggregate to form a large cell mass called plasmodium, not a malaria parasite. The spores produced by plasmodium are very tough and survive even very harsh conditions. Nucleonoids, they lack cell wall but have a tough covering of proteinaceous pellicle. They possess two flagella, one short and other long. They behave as heterotrophs in absence of light but possess pigments similar to that of higher plants for photosynthesis. Kingdom Plantae. The kingdom is dominated by autotrophs. It also includes some semi-autotrophic members. Now, the insectivorous plants are like Venus flytrap, the pitcher plant, bladderwort, as well as heterotrophic parasite members like Cuscuta. Members of this kingdom containing chlorophyll. Cells have cell wall alternation of generation, that is life cycle, has two distinct phases. The kingdom planting is divided into on two major groups: cryptogams and phanerogams. We will study this kingdom in detail in the next chapter. Kingdom fungi. These are eukaryotic or heterotrophs, showing extracellular digestion. They are found in warm and humid places. They have simple body, which may be unicellular or made up of long thread-like structures called hyphae. Now, large fungi such as mushrooms have a compact mass of cells. Unicellular organisms have a have a protoplasm with many nuclei. Example: Rhizopus, Saccharomyces, is unicellular fungus. Filamentous fungi consist of body called mycelium, in which hyphae are present. The hyphae may be with septa or without septae. They may be uni or multinuclear. The non-septate multinuclear hyphae are called conocytic hyphae. The cell wall is fungi. And fungi is composed of chitin, the polysaccharide or fungal cellulose. The fungi exhibit heterotrophic mode of nutrition. Mostly they are saprophytic. Some are parasitic or predators. They reproduce asexually as well as sexually as well as asexually. Asexual reproduction takes place by fragmentation, fission, and budding spore. Some fungi are symbiotic, either live with the algae as lichens or as mycorrhiza in association with the roots of higher plants. They are useful as well as harmful. Mushrooms are consumed as food. Yeast is used as bakery, and brewered penicillium, a fungus, is well known as antibiotic production. Harmful fungi causes diseases in plants and animals. Example: Puccinia. Now, the fungi are further classified on the basis of their structure, mode of spore formation, and fruiting bodies as follows. Phycomycetes. These are commonly called algal fungi. Mycelium is made up of septate conocytic phase hyphae. They are commonly grow in moist and damp habitat on decaying organic matter, as well as in aquatic habitats, or as parasites on plants. Example: Mucor, bread molds, albugo, parasitic fungus on mustard. Ascomycetes. These are called as sac fungi. These fungi are mostly multicellular. Rarely unicellular varieties include yeast. The hyphae are branched and septate. The sac fungi can be decomposers. Parasites are coprophilous, grow on dung, morels, and truffles. Varieties of Ascomycetes. These are called as sac fungi. These are fungi are mostly multicellular. Rarely unicellular varieties include yeast. The hyphae are branched and septate. Sac-like fungi can be decomposers. Parasites and coprophilous, grow on dung. Morels and truffles are varieties of sac fungi that are consumed as delicacies. Now, neurospora and useful in the genetic and biochemical assays. Example: Aspergillus, Penicillium, Claviceps, Neurospora, and Saccharomyces. Example: figure Aspergillus 2.10. Basidomycetes. These are commonly called club fungi. They have branched septate fungi hyphae. Example: Agaricus, mushrooms, Ganoderma, bracket fungi, Ustrilagos, muds, Puccinia, rusts, etc. Deuteromycetes. These are called imperfect fungi, which are known as reproduce only asexually. Example: Alternaria, Coletotrichum. Kingdom Animalia. Members of this kingdom are heterotrophs, adapted to holozoic nutrition. Most of them have a capacity of locomotion. They are multicellular eukaryotes where the cells lack chlorophyll as well as wall. Growth is determined, follow definite pattern. In chapter 4 we will study about kingdom Animalia and its further classification. Viruses. Viroids are groups of acellular organisms that are not included in Vitaca's 5 kingdom classification. Acellular organism. Viruses were named to show by Louis Pasteur, now considering the meaning venom or poison. These are obligate parasites were given the name virus or by M.J. Bajrank after agarugel, thus being infectious soluble agent. He called the filtrate as a contagium vivium fluidum. It was scientist Stanley who demonstrated that for viruses are inert outside the most host cell and can be crystallized. They are made up of proteins. Viruses lack their own cell machinery. They have protein coat capsid around nucleic acids and thus considered to be a cellular organism. Viruses are inactive outside of host cell, but once they enter their specific host cell, they take charge of cellular machinery of host cell and duplicate themselves. Viruses thus can be called infections, nucleoproteins particles. Types of viruses. As per genetic material, viruses are grouped as DNA and RNA viruses. Tobacco mosaic virus TMV. Figure 2.13. Protein coat called capsid is made up of smaller units are capsomers. Capsomers are arranged in polyhedral and helical forms. Capsid protects genetic material. The genetic material in viruses is either single-stranded RNA or single or double-stranded RNA or double-stranded DNA. Viruses that infect bacterial cells are called bacteriophages, which normally have double-stranded DNA. Viruses cause disorders like leaf curling, yellowing, mosaic formation, etc. In plants, you have heard of foot and mouth disease in animals, or swine flu, which are viral diseases. Smallpox, mumps, herpes, or common cold viruses are the causative agent of many diseases in humans. The list includes AIDS too. Viroids. Potato spindle tuber disease was found to be caused by a single-stranded RNA which lacks protein coat. Theo Danier in 1971 reported that this is low molecular weight RNA and smaller in size than viruses. These infections RNA stands are called viroids. Figure 2.14 Bacteriophage. तर एक्सरसाइज आपण नंतरच्या व्हिडिओमध्ये पाहणार आहोत ज्या आपला बायोलॉजी केमिस्ट्री आणि फिजिक्स याचा महाराष्ट्र स्टेट बोर्डचा सिलेबस कंप्लीट झाल्याच्यानंतर आपण एक्सरसाइज पाहणार आहोत. तरी तुम्ही या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला सबस्क्राईब करून ठेवा. प्लीज लाईक शेअर अँड सबस्क्राईब. थँक्यू सो मच फॉर वॉचिंग.